Today we're going to be covering a few chapters, I'd say, of research from the last 10 years of my career, really focusing on how modern beekeeping interplays with the virological health of our bees and some lessons learned on Varroa biology that I think would be useful to highlight given their recent invasion here into Australia. So before I get started, I always like to acknowledge the slew of funders that support my work back at the University of Georgia, including our Farm Bureau, USDA, which is our equivalent to DPI over there, Project APSM and the National Honey Board, which are our main beekeeper groups that fund my work, as well as Bayer and FMC. And in particular, this is a little bit of a brag from me, but I have a fantastic amount of support from my local beekeeping association, the Georgia Beekeepers, who have this very flashy license plate that they sell, where members of the public buy it to put on their car and support honeybee research and beekeeper education. So I'm going to take a fast step back to discuss some topics that will not be news to anyone in this room, but shaped a lot of my early career, where it's well known how important honeybees are for most of our pollination industries, and this is especially true with changing agriculture and will continue to be true, even as new technologies come in that might lead to fruit and vegetables that maybe don't require pollination as much. Many of our seed and nut and oil crops are always going to require insect pollination. And as farming continues on its trajectory towards large, intensive, monoculture-based farming, we need to be able to bring additional pollinator inputs into those environments, and really the only way to do that is with managed bees, and most of those currently are honeybees. In this way, the demand for commercial beekeeping is also part of the problem with respect to bee health. The environments that our bees are required to act in as part of their agricultural support for growing crops are not friendly environments for pollinators as a rule. They'll have limited diversity of nutrition in these monocrop farming contexts, high agricultural inputs in terms of chemistry, fertilizers, pesticides, whatnot, and just the stress of driving them around and moving them typically takes some small amount of toll. So we have this list of agrochemicals, habitat and forage loss. When I was growing up in the north of England, first learning to beekeep, we saw huge tracts of farmland, all converted over to canola, for instance, which can be a little bit of a pain for beekeepers because, as we all know, it crystallizes extremely readily in the frame, which was the bane of my existence as a late teenager. But these same fields are what drive the demand for the business that many of us are engaged in and what has ultimately funded my career so far. In the US, this has led to this widespread phenomenon of migratory beekeeping, whereby our bees are packed onto trucks and moved around the country in a similar fashion that you're all very familiar with over here as well. Much of my early work on honeybees focused on pest and infectious diseases and intersecting that with this slew of stresses that their agricultural environment brings to bear on them. And this was an idea that was first kind of posited by my former bosses, and so we're going to start by discussing some of my PhD work, looking at how all the diseases that afflict our honeybees might be altered in terms of their spread, their epidemiology, based on this industrial transition that we've seen in the last 100 or so years from traditional, small, local beekeeping to large commercial migratory operations. And I actually have good things to say, so just bear with me. Sounds all doom and gloom, but that's part of the storytelling here. So they're deeply tied to our modern agricultural system, and that changing environment that they've experienced over the last couple of centuries reflects also changes in their management. In my early work, I did some virological analysis to compare the health of colonies sourced from sort of 3 different honeybee backgrounds. Feral honeybees, which we caught in swarm traps from America's largest blackwater swamp. You can ask me about my snake and spider stick, which normally is like quite impressive to the American audiences, but that's probably just something every Australian household has, right? So we sourced honeybees from the middle of this swamp, which were very much a feral population, isolated. Beekeeping is not allowed to be done on that land, and so they were a true sort of wild equivalent. Traditional beekeeping, where this is small-hold, non-migratory, kind of backyard sideliner industry, and then the kind of high-intensity industrial commercial stock. We maintained apiaries that were separated from one another so that they couldn't cross-contaminate, but otherwise managed them identically for a year. Then we essentially biopsied them and looked at their viral levels. And I'm going to give you the very short takeaway here. What we found was that the feral bees behaved quite strangely. They had the highest viral levels for some viruses and the lowest virus levels for others, which is already contradictory to what my bosses had hypothesized early in my career, that the feral honeybees should be the strongest and healthiest because they're being selected for resistance to these pathogens. That was contradictory to what we ended up finding, and we'll talk about that a little bit more later on in this lecture. But what was very apparent was that in every instance, on average, the bees sourced from the large industrial commercial populations were sicker than their counterparts that had been managed in a more smallhold traditional fashion. And so reflecting on what could be driving these differences that we observed in the American beekeeping industry, whereby the style of beekeeping can be linked to how sick these bees are, at least in terms of their viruses, it's not necessarily that the beekeepers themselves are at fault. The actual spatial arrangement of the colonies, the crowding of them, yields potentially much easier conditions via which infectious diseases can spread. Genetic diversity of bees is critical to limiting the ability of these infectious diseases to spread within a colony, and so if the commercial operations are of more standardized background, then perhaps the smallholders, that might play a part. Their immune function is closely tied to the nutritional provision as well as exposure to agrochemicals, and then there's also some pathogen factors as well, or parasite factors. Things like strain-specific matching, whereby if you're moving your honeybees around a continent, they are just simply encountering a greater diversity of infectious agents, and so it's more likely that the one that's good at infecting them is going to be encountered by them. They're just sampling a greater number of parasites or viruses, and they're able to then be more severely infected compared to ones that stay in one place and are just simply exposed to fewer infectious agents. I think anyone that's had small children in their household is really familiar with that idea of how sick you get just by them moving around and acquiring far more bugs than we might otherwise like. So the first question that I sort of set out to answer earlier on in my career was whether the spatial arrangement, and by that I mean the crowding of honeybees into these massive almond pollination apiaries or onto trucks, has led to more deadly parasites. And again, this was an idea first posited by my former bosses. This leans on a whole field of evolutionary infectious disease ecology, which I'm not going to explain in great detail, but just take it from me that we have abundant evidence from bacteria to moths to theory that I've published. Do not go and look up this paper, please. I can barely bear reading it, and I wrote it. So of all my papers, don't go read this one. But we have a huge amount of evidence showing that as populations become more mixed, their diseases become more deadly. And there was a lot of concern in US beekeeping that as more and more beekeepers contributed to the California almond pollination, that mixing and exchange of diseases was yielding more deadly variants that were then spread back across the continent. And so I asked the question, how differences in beekeeping regimes change epidemiology and transmission of these parasites. Really, this is industrialization that we set out to study at the apiary scale, so kind of individually managed by beekeepers, because we can do experimental tests on that, and it's something that beekeepers have control over. You don't really have much control over what crop is being planted across the country that you're servicing. You don't have as much control about what they're foraging on or what chemicals they might get into, but you do have control about how you arrange your apiary and how many colonies go into a single spot. This was work that was published some years ago now. It's getting a little bit old, but it ultimately just addresses the very simple question of, does crowding colonies lead to more prevalent diseases in our honeybee stock? And normally when I'm explaining to other scientists, I have to tell them how instances like this lead to high levels of drift of honeybees between their colonies, drones in particular, and not only often more sick because they're immunologically kind of feeble, but they're also stupid, and so they're very, very likely to move colony to colony and spread those diseases. Beekeepers themselves might also move frames of brood between colonies, and that's another opportunity for abundant disease transmission. And so we did some fancy mathematics and some computational simulations to model the spread of test diseases within apiaries, kind of in a computational model, almost like a video game. And we recovered, first of all, a very simple and obvious fact that the more easily a disease transmits, which is this R0 number on the x-axis here, the more bees get sick from it. So the better a disease spreads, the more bees get sick. Simple, easy, both our models agreed on that. What we then started to focus on instead was altering the arrangement and style of beekeeping that we were simulating in these models. And that allows us to start studying how the pathogen biology alters epidemics that we're simulating in beekeeping, but also how the honeybee ecology or the beekeeping management matters. The way we addressed this was to compare a kind of very small traditional 9-colony apiary with very low rates of movement of bees between those hives, compared to a much larger sort of hypothetical commercial apiary of many hundreds of colonies packed into a tight grid with abundant opportunity for bees and infectious diseases to move between those. We simply said, how much worse are disease outbreaks in the left traditional apiary compared to the right, more modern commercial style apiary? So again, moving from left to right, how much more severe is the disease burden in our colonies? We did this by measuring how much more effectively these make-believe kind of model diseases can spread in that commercial apiary compared to the traditional one. And once we had this piece of information, we could link it back to our original graph mapping how transmissibility relates to disease burden. With those two pieces of information, we can then ask, if a beekeeper were to convert an apiary from being small, traditional, spread out, kind of the idyllic beekeeping we've been doing for hundreds of years, to a more modern commercial apiary, how much more sick would those bees be? And the answer actually was very, very little. Turns out that crowding cannot explain, no matter how you twist the maths, no matter how much you try and get these models to yield a bad epidemic. There's almost no difference between the expected disease dynamics in a very traditional, old-fashioned form of beekeeping compared to our modern large-scale commercial beekeeping. It took me a while to persuade my colleagues around to this, and this is an example just showing where we'd expect kind of Nosema to sit in an example colony system. Apologies, I was ranting and raving at the pub last night, so my throat's a little tired. And I think really this comes from a mistake in how we perceive old-fashioned beekeeping to be low density. An individual colony still has tens of thousands of individuals packed into that tiny hive, and even if you've only got 5 or 10 of them next to one another, that's still 100,000 individuals, and it only requires them getting confused as to which their front door is once or twice for diseases to spread quickly throughout an apiary. And what we essentially found was that commercialization or industrialization of beekeeping can't have made disease outbreaks worse, as there was no room for them to get worse. Basically, if a disease gets into a hive, it's getting into every colony in that apiary, no matter how little drift you try and mandate there is in the colony. If you're not changing brood or whatever, it is still going to reach every single bee because they already live in such highly dense societies compared to most of the rest of our examples in nature. And so, fortunately, I get to tell you one thing you don't have to worry about from a disease perspective, and that is that packing more colonies into your apiaries or putting bees on trucks and moving them isn't meaningfully exacerbating disease spread. Basically, it's impossible to stop disease jumping colony to colony if they're all in the same apiary, at least from the modeling work we've done. So crowding doesn't seem capable of meaningfully exacerbating infection in honeybees, Managed bees live in such dense societies that if a pathogen gets into an apiary, it's getting to most bees regardless. Fortunately, at the same time as we were doing this modeling work, a lot of the early breakthroughs in more kind of sensitive and accessible virology for bees were being published. And so these papers by mostly European and some American authors that were looking at the individual infection rates of viruses in bees found that anywhere between 3 out of 4 to 100% of tested bees always had, for instance, deformed wing virus, backing up our finding that between 70 and 100% of bees should be infected with any given pathogen if it's circulating in that population at all. So that meant I could start eliminating some of the factors that underpin that early epidemiological question I asked of why are our commercial bees suffering these worse infections. And I think part of one of the lessons I learned was that in my field of the ecology and evolution of infectious disease, or EEID, that I was trained in, we're really trained to think about animals being infected or not. And that comes from kind of vertebrate veterinary medicine, where our immune systems operate very differently to insects. And I think in honeybees, we need to think a lot less about binary infection or the presence versus absence of a parasite or a disease, and think much more carefully about the severity of that infection. A hypothesis that I often work with when looking at honeybee diseases is that just about every colony has just about every disease, at least in the US, if you have a sensitive test that you put in there. If you're good enough at getting material out of the colony, you can just about turn up everything. What matters is how the health and vitality of those bees allows them to suppress those infections that are always waiting in the wings. That's a very different way of thinking about animal medicine to what we're typically trained in elsewhere in other livestock systems. So to go back to this question of what was the cause of these consistent differences in viral infection between our commercial and non-commercial bee stock, we can look back to some of the host factors that I originally introduced, things like the honeybee's immune function, which is closely tied to their environment, as well as their genetic diversity. So we're going to go on and talk about those for a little bit now. So we know that wild bees have been declining alongside our managed bees because of this list of problems that they encounter, and these things all interplay together with one another. Toxic chemicals impede bees' foraging ability, that in turn limits their ability to furnish themselves with abundant nutrition that can immunosuppress them, leading to more severe parasite outbreaks. And this idea of these interacting stresses is something we're going to spend a little bit of time on, where all the things bothering our bees, be it pesticides, be it disease, be it limited nutrition, all exacerbate one another. Bees that aren't fed well get more sick. Bees that are immunosuppressed because of a toxin in their environment get more sick. Sick bees aren't as efficient at foraging, and they are less capable of bringing large nectar crops back just because the running a fever of an infection is more energetically demanding on them than if they were perfectly healthy. And so as we're thinking about managing the infectious disease burden in a colony, not just whether or not a parasite is present, not just whether or not a disease is there, but how severe is the disease in that colony. Really, it's about taking a step back and looking at all the environmental factors that are underpinning the health of those bees and their own ability to limit varroa or viruses or whatnot that's affecting them. And there's a whole slew of papers with these increasingly insane diagrams by my colleagues detailing all the interactions that we might want to study if we're truly going to understand what drives honeybee infectious disease. So similar work's been done elsewhere with similar findings on these interactions, and I'm going to talk about some of how this plays into common ideas that I do want to also maybe dispel. One thing that I do want to highlight is going back to that finding I had that our feral bees were not necessarily healthier than our managed bees. There were numerous viruses that they had much higher viral loads for than our even commercial stock. A similar result was found a couple of years later up in the north of the US in Pennsylvania, where they pair-matched kind of unmanaged feral colonies or populations of honeybees next to ones managed by beekeepers, and they also consistently found that the feral bees were sicker. So there's nothing wrong with large-scale beekeeping implicitly from a bee health perspective, so don't fall into the trap of blaming modern beekeeping for these problems. It's not particularly defensible. Feral bees are also not inherently healthier than managed bees in some like airy-fairy Gwyneth Paltrow jade egg goop nonsense. Bees with someone looking after them are healthier than bees who are left to do it on their own. And I think that should make us as beekeepers also feel good that we are in fact helping the creatures we are charged with looking after. So pat yourselves on the back for that. Rather, the landscapes our bees exist in, as well as some of the choices we make about their genetics, are ultimately, I think, what we have control over that goes into determining their health and their parasitological status. So one thing that is of particular interest to me that few other people talk about, because it's a nightmare to work on, is the internal genetic diversity in colonies and how that determines how sick they are. And this is a recent paper that we published that builds on a long-standing legacy from Tom Seeley's lab and Dave Tarpey and others, looking at the very obvious link, whenever you bother to search for it, that queens that were more polyandrous and more diversely mated in the choice of drones that they were given always head colonies that are more robust and resilient in the face of parasites and infectious disease. And I highlight this because there is a huge amount of effort and attention put into breeding bees with these resistance alleles, which is worthwhile and we should be doing, but we don't want to box ourselves into a corner where we're eliminating a bunch of ubiquitous genetic diversity that's not only useful for the future of our beekeeping, because we can only select from the diversity that's present, but the presence of a diverse set of worker genes in and of itself is already helping reduce infectious disease burden. So we've done some work recently where we AI'd a bunch of queens with very controlled mixes of sperm, where we did everything from kind of single inoculation right through to mixes of 32 drones. And really the question we were asking here is whether or not diversity in and of itself, so the diversity of genetics in the spermatheca, is what's important for this consistent finding that more genetically diverse colonies are more robust, or whether it's a sampling sort of bias, and that simply queens that mate more are more likely to have mated with a drone who possesses a rare allele that confers some degree of resistance in the form of a hygienic behavior amongst a subset of the workers. So we, not me, because I'm really piss poor at artificial insemination of queens, more talented people than me did the AI. We monitored these colonies for a long time, a couple of years, and the consistent finding we uncovered amongst these experimental apiaries was that the more polyandrous a queen, i.e. a queen with more diverse sperm set inoculated into her, and this all, I'll just make sure to state this, all queens received exactly the same amount of sperm. It was simply the diversity and composition of that sperm set that she was inoculated with. Were more likely to survive overwintering. They headed colonies with smaller Varroa populations in absolute terms, and headed colonies with smaller Varroa populations in a per capita sense. And so just by having more diverse colonies, we found this explicit link between how sick they got from Varroa. And this shows those results over time with the negative correlation there between their polyandry rank and how much Varroa there was in the colony. An interesting finding that came out of this as well was the Varroa populations grew more slowly in these more diverse colonies. So the Varroa themselves seemed to show either higher death rates or lower fecundity when faced with a more diverse population in which they were trying to grow. We wondered if this might be underpinned by this sort of rare sperm hypothesis that by inoculating with a greater number of drones, we were more likely to instill some genetics in that colony that led to a couple of workers having these sensitive behaviors, behaving almost like colony white blood cells, the so-called rare sperm hypothesis we were dealing with. And we found absolutely no evidence for that. There was no individual drone that seemed to impart resilience to the colony in terms of Varroa resistance. Instead, it was just that in every case, the more diverse the genetics of the colony proxied here by the diversity of the sperm delivered into the queen, the fewer Varroa were in that colony. And so queen health is another major pillar, we all know, of beekeeping, but this is an explicit link between the mating quality of that queen and how likely it is that Varroa are going to be able to easily grow inside that colony. Can I get a time check? 5 minutes? Cool. We'll talk about one final experiment then that builds on this, where we also asked if genetic diversity for the colony is so critical to their epidemiological status, can it also mitigate some of the impacts of toxin exposure in the form of pesticides? So we know that increasing genetic diversity makes for stronger colonies. We know it specifically helps suppress parasites. Can it also enhance their detoxification abilities? This builds on some work we did a few years ago in Iowa, where we kept bees along a really strong agricultural gradient and unsurprisingly found that the #1 cause of death in these bees was Varroa, but that Varroa level was very explicitly predicted by the agrochemical profile that we detected in the colonies linked to what farms they were adjacent to. I'm not going to go into much detail on that because I didn't lead the work, but it's there if anyone's interested in it. And so we were wondering if more diverse colonies have better detoxification skills, essentially was the question we asked here. And rather than doing AI in this case, because as I said, I'm not very good at it, we did brood transplantation between queens of known genetics in order to artificially elevate the genetic diversity of colonies by exchanging workers so that they had more diverse populations for the 6 or so weeks after those exchanged brood frames emerged. We then force-fed them pollen with field-relevant pesticide doses. So here's just a schematic of that, where we have either genetically homogeneous versus genetically heterogeneous colonies, and then pollen that was either clean, kind of NMR tested, versus pollen that was spiked with field-relevant doses of thiamethoxam and clothianidin, which are both neonicotinoids. We did this across 3 big locations in the US, 3 different states, and assayed about 20 different parts of the colony's phenotype. At the same time, some phenomenal work came out of Italy that I was ignorant of until the paper was published, where they raised Varroa on pupa in the lab and showed that pupa that had received a sublethal dose of neonicotinoids during their larval stage were weaker at protecting themselves from Varroa, and that manifested in that an individual foundress had more daughters successfully when she was able to feed on pupa that had been exposed to pesticide versus not at the larval stage. And this was shown to be mediated via the honeybee's immune system, where these sublethal neonicotinoid doses fed to the larva hamper the honeybee's immune system, and that leads to easier time for the Varroa. And so to cut a long story short, before Martin drags me off stage. Oh, 15 minutes? Oh, we've got loads of time. Okay, never mind, you're all not free of me just yet. I thought that was going to be the 5-minute mark. Perfect. Okay, so the major finding we had from this, where we force-fed pollen to these bees that were either genetically diverse or genetically less diverse, was twofold. One, we found absolutely no evidence, unfortunately, that genetic diversity helps buffer colonies against the effects of these toxins. We couldn't find that the more diverse the colony, the better they were at ridding themselves of this spiked pollen, and that they weren't mounting a more effective toxicological response, which was disappointing. And we're not really supposed to say that as scientists, because we're supposed to be unbiased and objective about our experiments, but I do think it's fair to say that I was disappointed we couldn't come up with at least one way to help our bees deal with these pesticides in the environment. The second major finding was that of the 20 or so colony health metrics that we measured, and we looked at diversity of pollen and amount gathered, we looked at honey stores, we looked at aggression, we looked at larval rearing success, queen failure rates, name a colony health metric, and we probably measured it. The only negative effect we could find of force-feeding this pesticide to our bees was that their Varroa levels were much higher if they had been fed the pesticide. And so the primary mode of action of this pesticide on affecting colony health and survival is levied via Varroa. It's not directly killing the larvae, it's not suppressing the brood area, but it is hampering their immune system, leading to more severe Varroa parasitism and consequently, eventually, colony death and collapse. That sounds scary, right? Because the more toxins our bees are exposed to, the more vulnerable they are to Varroa, but I actually think there's a bit of a silver lining to this result, and that is that Varroa are something we can handle. We have treatments for them. We have a whole tool belt of ways in which to try and keep Varroa levels low. If the primary mode of action of these neonicotinoids in compromising honeybee health was kind of direct toxicity, all we can do is try and beg our bees not to go feed on crops that have been sprayed with them, which we know doesn't work. But if the negative impact of those agrochemicals relies on the presence of a parasite to exploit their immunocompromised state, that's a bit of a chink in the armor there that we can attack. Because if we can eliminate or keep Varroa very low, the impact of these chemicals on our bees is much, much less severe. And I think that's something that we really need to kind of cling to, is that if we want to help defend our bees against toxin exposure, eliminating these additional stresses that can capitalize on that immunocompromising nature of these chemicals is the critical way in which we can kind of intercept them. Just to mention, so that no one questions the kind of power of the experiment, we did find that increasing the genetic diversity of the colonies had other direct beneficial effects. Larvae always survive at higher rates in more genetically diverse colonies. So we did find benefits of the genetic diversity. We just didn't find that it mitigated directly the negative effects of the pesticide. So no direct leads on reducing pesticide damage, but some information that better informs us as to how pesticides end up killing colonies is often by working in concert with these other actors. So, applied takeaway from this: Varroa management within operations leads to very different outcomes based on location. You might have two apiaries that you're treating exactly the same from your Varroa management perspective, but if one's getting into a neonicotinoid-laced field, something like canola, and the other one isn't, you're going to see far worse Varroa mite levels in the colonies that are being exposed to these agrochemicals. And that can be very frustrating as a beekeeper when you have bees that are of the same origin, you're managing them the same, you're feeding them the same stuff, you're treating them exactly the same, and some yards are just always full of mites and some aren't. It's often because of these indirect effects of the local environment that that apiary is placed in. From a broader policy-facing point of view, I think we aren't doing, as of yet, the best job we might when we're assessing the toxicity of these agrochemicals on honeybees. If we're just looking at it in the lab without the presence of these additional parasitic stresses, we're probably underestimating the effects of these pesticides on our bees. And so in a perfect world, we'd assess pesticides very holistically, but I do think with easier and easier technologies looking at things like transcriptomic responses in bees, so how their immune systems are regulated based on the dosing of these toxins, we might be able to get a better idea of which of our agrochemicals might be having these indirect effects ahead of time.